Alright, this video is going to cover Objective 3 of Chapter 10. We're going to be taking a look at the myofibrils. So as we're getting ready to learn about contraction, we have to understand about the protein filaments that are going to be involved in the contraction. And we're going to find those in the myofibrils. And so we're going to just start by identifying what is a myofibril. Okay, so this was actually mentioned in the parts of a muscle cell. But this is something we haven't seen before, but this right here represents one muscle cell. And so one thing you have to understand is that we have smaller units inside of bigger units. And so here we have one muscle fiber, and as you can see, there's lots of these little cylindrical tubes within that muscle fiber. And then there's going to be lots of these muscle fibers in a fascicle, and then there's going to be several fascicles in a muscle. So we kind of have this smaller to larger tubule type system in terms of the muscle structure. Now inside of that muscle cell or that muscle fiber, we're going to find lots of these myofibrils. Okay, so they're these long tubes. And so they're a long cylindrical organelle. And these are the organelles that contain our actin and myosin. These are going to be the major players when it comes to our contractions. And so we need to understand about the myofibrils before we can move forward and talk about the contractions. Now, in addition to those myofibrils, you can see that they kind of fill up the bulk of the cell, and so they kind of squish the nuclei over to the side of the muscle cell. You also have mitochondria throughout to provide energy. You do have the sarcoplasm, which is going to be just your basic cytoplasm. And then you have this specialized smooth ER called the sarcoplasmic reticulum. You can see it's kind of this tubule network. And going to the sarcoplasmic reticulum are these transverse tubes called your T-tubules. So they kind of surround each of those myofibrils, and then they're kind of connected to that sarcoplasmic reticulum. So we're going to see those terms coming up again here in a little bit. And so the myofibrils are those thread-like structures. There's a whole bunch of them all inside of the muscle cell. They go from one end of the muscle fiber to the other end of the muscle fiber, so they're these really long tubes. And within the myofibrils are these bundle of proteins called myofilaments. Sometimes it'll just simply say filaments, okay? So like here it says actin filaments, myosin filaments. These are your myofilaments or your muscle filaments. And so actin are the proteins that are thin. A lot of times you'll just see them described as the thin filaments. And then myosin are the proteins that are thicker. And you'll often see them described as your thick filaments. And so we're going to take a little bit of a closer look into each of these proteins. Now keep in mind they are proteins, and proteins are always very specific about what they do. And so when it comes to your actin, you actually have two strands of what we call the F-actin. And it kind of forms this helical structure. So these little blue dots up here. Okay, so there's kind of the basic protein chain. And then we have two other types of proteins. We have tropomyosin, which is kind of like this band here. And then we have troponin, which is this little group of proteins. And so the F-actin forms that little double helix shape. It's composed of the monomers. So the big helix shape would be a polymer made up of many smaller units. And so those single units are called your globular actin or your G-actin. And what's special about them is that they have an active site that myosin can bind to. This is going to be really important when we talk about muscle contraction. So on the actin protein, on these little monomers, we have a location where myosin can actually bind to it. And we'll kind of see that when we get to contraction. Now, in addition to those, the F-actin, those long kind of double helical structures, we also have tropomyosin, which is this kind of elongated protein. It kind of looks like a rope that kind of twists with that helix. Now, what it does is it actually covers those active sites, okay? So each molecule kind of covers up that binding site for myosin. And then troponin is going to be this little group of three proteins here. It's got different subunits. And so one subunit can bind to actin. This is where the troponin actually attaches to that F-actin region. One of your subunits can bind to tropomyosin, that rope type structure. This is the one that's going to be important in muscle contraction. And then we also have another one that can bind to calcium. So this is going to be kind of our trigger. Calcium is going to bind, and then it's going to cause a change in that troponin. And then this is all going to be important in how muscles contract. So you have troponin I, that is an I, troponin T, and troponin C. All right, now on the myosin filaments, these are my thick filaments. So they actually have many of these myosin molecules, and each myosin molecule kind of has this double head structure. All right, so this is one myosin molecule, and each myosin molecule is going to have the two tails that are called heavy chains, and then they're kind of wrapped together, and then they're going to have these two myosin heads. Now, the myosin myofilament as a whole contains about 300 of those myosin molecules. So you can see that there's a whole bunch of those little golf club shaped structures all put together, and their heads project towards each end. So you've got some going this way and you've got some on this end. And so all together that makes a really thick type of filament. So your actin were your thin filaments and your myosin are your thick filaments. And so what we need to understand mostly about the myosin are these myosin heads, because they kind of stick up, they kind of are coming away from that myosin kind of filament, and these are going to be the parts that are going to bind to those active sites on that actin molecule. So they're attached by the tail portion, and they have this little hinge region which is going to allow those myosin heads to kind of move, to kind of bend and straighten. There are also going to be enzymes that are going to break apart ATP. So we'll talk about the ATP portion when we get to muscle contraction, but they're able to break down that molecule and release energy. And so that's going to help provide energy for them to move at that hinge. So when you're looking at your myofibrils as a whole, there are these really long tubes like this, okay? And within these long tubes of the myofibrils, there are multiple sarcomeres, okay? This is essentially the smallest contraction unit. So we're going to look at muscle contraction in terms of one sarcomere, okay? But you need to understand that there are lots and lots of sarcomeres along that myofibril. All right, so they're going to work together and they're going to work in unison, but there's many contraction units, little individual contraction units within each myofibril. And so each sarcomere is going to consist of the actin thin filaments and the myosin thick filaments and the proteins that kind of regulate them. There are thousands of these on a myofibril. Okay, there's thousands of sarcomeres on one myofibril. Now the way we kind of identify them is they are enclosed by what we call Z-disc, or sometimes they're called Z-lines. And that's just pretty much a protein that goes across, and that's where your actin proteins are going to attach. And so here you have your Z-lines or your Z-disc, okay? And then your actin is going to bind to that Z-disc. There's my thin filaments. Same thing on the other side. Now earlier we talked about striations in video 1, about how that skeletal muscle and cardiac muscle were striated. And what gives the appearance of those stripes has to do with these actin and myosin. So you can see here that in this region we only have those thin filaments. Okay, we only have the thin filaments here. And these are what we call our I bands. Okay, these are going to be the lighter color stripes in our striations. And then you can see here where you have these thicker filaments, you have that A band, and this is where they're going to appear darker in terms of the striations because those filaments are a lot thicker. The M line represents the middle of the sarcomere, and what's going to happen is in a contraction, these Z disc are going to move toward that M line. And then the H zone is this area where you only see the myosin filaments. Okay, so you have some overlap here, some overlap there, but in the middle, you do only have myosin filaments. All right, and we're going to end that video, and we will pick up on the last video for this particular PowerPoint on contraction.